Namaskar दोस्तों, welcome to channel Toxicology on Board. Today in this video we'll be discussing another DABT topic that is in continuation with the Clayson short textbook, series of questions. In second chapter, that is Principles of Toxicology, we have total 10 questions are there. In this video I'll be discussing question number 1 to 5. Okay, let's directly dive into the question. So the question is, the 5 identical experimental animals are treated with 1 mg of one of the following toxins. Okay, the dose is 1 mg. So we need to identify the animal tested with which toxin is most likely to die. So the question, the concept, what we can learn from this one is about acute toxicology studies, that is LD50 value. So we need to compare the LD50 values of all these different 5 different chemicals, that is ethyl alcohol, botulinum toxin, nicotine, ferrous sulfate, picrotoxin. Okay, this where you can learn. There is a table which is given in table 2.1 in this cassette and all essentials of toxicology, third edition, page number 7. It is given. Also, in ninth edition textbook, bigger textbook is there, no? In that also this same LD50 table will be there, where is a comparison of this various LD50 ranges. So you can see that ethyl alcohol, its LD50 is 10,000 mg per kg. When it see another botulinum toxin, which is the most potent, okay, the LD50 is 0.000001 mg per kg. It is in microgram level, okay, or very at very low dose only. This is going to cause a lethal effect. So that you have to compare. So these values, no, they are directly given from the table. Ethyl alcohol 10,000 mg per kg, botulinum toxin 0.00001 mg per kg, nicotine, see nicotine 1 mg per kg, ferrous sulfate. Okay, ferrous sulfate they have given 1500 mg per kg, and picrotoxin LD50 is 5 mg per kg. The right answer will be green text, that is botulinum toxin, because we need to identify the lowest dose which is causing uh, 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 toxicity. So from this also we'll also learn, again it's a revision, already you people might be knowing. Let's again do a quick revision. LD50 stands for lethal dose 50, amount of toxic substance required to kill 50% of this tested animal population. Okay, this LD50 used to measure short-term poisoning. So repeated type of exposure that you cannot get it from this acute toxicology studies, single dose toxicology studies, or short-term studies. Okay, that you can from this acute toxicology studies, what you can get is LD50 value. Nowadays you can get MTD also, maximum tolerated dose. But I see certain pharmacology reviews in FDA pharmacology reviews in the single dose toxicology study also they are giving NOAEL. I am not sure what is, because to establish a NOAEL, you need to have at least 3 different doses. There you can establish a dose response curve. I am not no understanding how they are deriving, but I come across in the literature. But major thing I'm saying, a repeat dose toxicology study, you'll get NOEL, LOEL, MTD also. In the case of single dose toxicology, the endpoint what we are defining is LD50 as well as certain times MTD. Okay, the value is often expressed in mg, milligrams of substance per kilogram of animal body weight. So units are important. Oral is mg per kg, inhalation it is mg per liter or ppm, mg per meter cube, like that. And in case of that you will get LC50, lethal concentration of 50% population. Dermal also you'll get the units in mg per kg only, and that's all. Okay, let's dive in. In addition to, because other types of, whether it is ferrous sulfate, ethyl alcohol, all those in my channel I have discussed. But picrotoxin I haven't discussed. So by taking this question as an opportunity, we'll learn about picrotoxin. So picrotoxin is a crystalline plant compound that is a mixture of 2 molecules. So it contains 2 components. That is picrotoxinin as well as picrotin. Okay, these are the 2 important things. How the mechanism of action is, picrotoxin, it will block the chloride ion channels activated by this GABA and glycine. So you have to remember when GABA, what are the inhibitory neurotransmitters, stimulating neurotransmitter, you have to remember that is GABA inhibitory active will be there, no, that one. GABA and glycine chloride ion channels activated. So GABA and glycine are the neurotransmitters which will activate these chloride ion channels. picrotoxin is going and binding to that particular channels, ion channels, and blocking them. As a result, what inhibitory neurotransmissions, GABA inhibitory, glycine inhibitory in the central nervous system, and leading to excitation and conversions. Okay, GABA and glycine has to stop it by inhibiting this particular, by using this toxin. It is causing excitation, repeated signaling from one neuron to another neuron, because of which there will be convulsions and seizures, that you have to remember. Symptoms include headache, chills, saliva, vision loss, and conversions, as I said. No. Next antidote for this particular thing is piscrotoxin was used as a CNS stimulant and antidote for poisoning by several nervous system depressants, especially barbiturates. So there is a barbiturate poisoning, you are using picrotoxin as an antidote. And research tool, yes, picrotoxin was used as a research tool to block GABA A receptor and study their function. This was the important thing that we can learn from this particular option, picrotoxin. Next, we'll dive into the second question. The second question in the textbook was, place the following mechanism of toxin delivery in order from most effective to least effective. So, which we have to identify the route of administration fastest to the least. This is a basic principle, but still certain times we get confused even by repetitive reading also. Let's remember this one: intravenous followed by inhalation followed by subcutaneous, okay, subcutaneous, then oral, then dermal. So always remember, okay, 100% bioavailability, inhalation will be 80 or 90% bioavailability, subcutaneous, okay, insulin you are giving through subcutaneous, oral and dermal. Dermal, if the membrane, if your skin, epidermal, whatever the layers of your skin, they are intact, there is no compromise, the layers are not disrupted, then dermal will, the route of will be. That's why in LD50 values, if you see dermal, no, 2000, 1000, like that, you will be having the values mg per kg. Okay, so these are the various routes of administration. Next we have question number 3, that is, a toxin with a half-life of 12 hours is administered every 12 hours. What is the which of the following is true? So you have to remember, half-life is 12 hours, which is equivalent to next administered dose. So they actually, if you look into this textbook, no, Glaser 9th edition, page number 33, you have this figure 2.4. This particular question is matching with, here you see line B. This is the B line, and what does this line B in this particular graph states that you're taking time on x-axis and concentration at the target sites on the y-axis. Line B, it will say a chemical with half-life approximately equal to frequency of dosing. Here see, half-life is 12 hours and it is one next dose administration is also 12 hours. So this particular B line is there, no, it is matching with our question. Okay, example, 1 day. So we see what is the options we have. Chemical is eliminated from the body before the next dose is administered. No, still it is there. Okay, equal it is. Concentration of the chemical in the body will slowly increase until the concentration is attained. Yes, that is the reason. Here you can see B line, how the repeated administration it is, after first, second, third, fourth, fifth administration, it is reaching to the toxic response. A toxic level will not be reached regardless of how many doses are administered. This is absolutely wrong, because if you can see, after fourth, fifth administration, toxic level is attained even with this same half-life 12 hours. Next, we have acute exposure to the chemical will produce immediate toxic effects. That is not always true. After acute exposure also, certain times the adverse effects might shown later on, after a certain period of time, after a long period of the exposure time also, including in, if you see acute toxicologist LD50 studies, even you see, you will be observing 30 minutes, 1 hour, 2 hours, 4 hours, 8 hours, 24 hours, 48 hours, 72 hours. And there are historical also, lot of examples are there, exposure at one time, but the what that adverse effects has occurred at a long time, after the exposure has taken after a long period of time. So this D option is also not right. The next we have option E, that is, elimination rate of toxin is much shorter than the dosing interval. That's not true, no, because it is equal to 12 hours is equal to 12 hours. So option E is also not applicable in this case. So right answer will be option B. And by taking this question, we also try to learn this figure 2.4, what line A is saying, line C is saying. See, if you see this particular graph, no, it is a diagrammatic view of relationship between the dose and the concentration at this site under different conditions of dosing frequency and elimination rate. So dosing frequency is time points 1, 2, 3, 4, right? And if you look into line A, this dotted line which is there in the shaded region, blue region, no, a chemical with low, very slow elimination. Example, half-life of 1 year. Once you have administered, the chemical is being in your body for more than 1 year. For example, we have radioactive materials are there, some other type of drugs are there, electrolyte are there, which go and deposit in your bone. There is another drug is there. Like many examples are there whose half-life is more than 1 year. That's why they will be retained in this concentration range of toxic response only. Line B we have discussed. Let's see what this line C, that red color line is there, no? Rate of elimination faster than the dosing frequency. You have administered 5 mg of drug and half-life is, for example, 2 hours. The drug is going out of your body within 10 minutes or 15 minutes. Okay, so elimination half-life is faster than the next dosing frequency. But here they have given example 5 hours. So blue shaded region is representative of concentration of the chemical at the target sites. For example, our target site for this ABC was, for example, liver, you assume, necessary to elicit a toxic response. So try to read this graph, very important. Based on this concept, many times many questions have come across. So it is there in the page number 33. Then we are going into next question, that is question number 4, that is artiol. Artiol, again a repetitive topic of question, is the toxin found in poison ivy. Okay, must first react and combine with proteins in the skin in order for the immune system to recognize and mount a response against it. Artiol is an example of which of the following. So we need to identify this, either it is antigen, it is autoantibody, super antigen, apten, cytokine. The right answer is in the green text, that is hapten. So what is hapten? That we will also read. These are the hapten, these are the carriers. Once a hapten binds to the carrier, it forms a complex, that is complete antigen. As such, hapten will not have any toxic properties until unless it is bind to the carrier. So let us understand what is the definition of this hapten. It is derived from the Greek word haptate, which means fasten, to fasten. Smaller molecules that elicit an immune response only when attached to a large carrier. In this case, this is the carrier, large carrier, such as a protein. So, artiol will go and bind to a protein. The carrier may be one that also does not elicit an immune response by itself. The carrier might itself show certain immune response, or it might not show an immune response. But definitely, when a carrier comes in contact with a hapten, a complex will be formed, and that will definitely have immune response. The mechanism of absence of immune response may vary, involve complex immunological interactions, but can include absent or insufficient stimulatory signals and antigen presenting cells. So there is something called APC cells are formed. We'll see next slide. There will be Langerhans cells will be formed in. So let's understand this concept in detail, because this is a repetitive topic of question. So artiol hapten type for hypersensitivity reaction. In addition, I will also tell you to read all four. But I recently came across there are total 5 types of hypersensitive reaction: type 1, 2, 3, 4. Fifth one is also there. Read it. Okay, and how they are mediated: IgA, IgM, IgG, IgM, cytotoxic. These are the comparison use. Read. Then we have before the artiol. So once this poison ivy, a person comes in contact with this poison ivy plant, leaf or any other part of the plant which exude this particular poison, no, what happens, first of all you need to wash of that particular exposed region. Okay, washing of washcloth using soap and water. Okay, so whatever the unabsorbed part of the poison is there, it is weared off from your exposure region. But the one which has already been absorbed into your skin, substantial amounts of artiol may be absorbed within minutes. So the absorption time is very very small. Within minutes, some part of the chemical, that is artiol, might penetrate into your skin. Now next is, once artiol has been penetrated in the skin, attempt to remove it with the water is ineffective, because already it is absorbed. There is no point in again washing it. After being absorbed by the skin, this artiol is recognized by the our immune cells. Our body immune cells will be the defense mechanism. What it will do, it will start activating this immune system dendritic cells. Dendritic cells are there, also known as Langerhans cells, right? They will, this dendritic cells or Langerhans cells, they will identify this particular artiol, and they will, what they will do, these cells can then migrate to lymph nodes. Okay, here are the lymph nodes they can migrate, where they present artiol to the, they will come in contact with this T lymphocytes are there, okay, and thus recruit them to the skin. T lymphocyte cause pathology through the production of these cytokines. So the cytokines will start accumulating in that particular region, and it will cause cytotoxic damage to the skin. In this particular case, it is painful rash, blisters, and itching. These are the symptoms externally which you can see. This is a very beautiful diagrammatic representation is there. This is the uppermost your skin layer is there, skin surface. What is happening is artiol or any allergen which is coming in contact with the skin surface. What is happening? It will penetrate and it will reach this particular epidermal layer. No, in that Langerhans cells get activated, right? Then this will uh, come in contact with this lymph nodes, and there you will see this effector T cells, receptors, all this undergo elicitation phase. Okay, then lymphokine released by this effector T cells, killer cells, lymphokine release toxins that destroy artial allergen as well as skin cells. So this is the basic mechanism. Try to remember, and also remember it is a type 4 type of hypersensitivity reaction. Poison ivy, another diagrammatic representation is given. Okay, here you can see T cell activation, and you can see artial induced contact dermatitis and psoriasis. You can find it. And what is the chemistry behind it is that to cause an allergic dermatitic reaction, artiol, this is the basic structure of artiol. You can remember, like, it's a benzene with 2OH phenol catechol type of structure is there, no? It undergoes oxidation, and this OH bond is there, no? It will become ketone, oxidate to create double bond oxygen on the chemical, right? This particular thing, then it will come in contact with your DNA, okay, protein nucleophilic, to trigger a reaction within the skin. Okay, once it comes in contact with the protein, then only that complex will be formed. Mediated by acquired immune response, too small to directly activate immune response. That's why it has to come in contact with the carrier, reaches a certain proteins of the skin where it acts as hapten, leading to type 4 type of hypersensitivity reaction. This is very very important. Oxidation, protein nucleophilic, right, oxidate, a double bond will be formed, right? Next, we'll dive into last question, that is toxic chemicals are most commonly to be biotransformed in which of the following organs? The right answer is very simple question, that is liver. Try to know the list of the organs. Different liver is involved, kidney is involved, skin is also involved where excretion of drugs takes place, right? Sorry, not excretion. I mean to say biotransformation, metabolism. In the metabolism also you read phase one, phase two, and phase three. Methylation, conjugation, all those things you read. So this is a direct question. So most likely to be biotransformed by liver. Heart, lung, CNS, they are also correct only, but major, most likely, that is answer is liver. So hope from this question you were able to learn some of the few principles of toxicology. We have read about LD50, we have read about cumulative, how the repeated exposure, that examples, case studies you have seen. You have seen about artiol type 4 hypersensitivity contact dermatitis reaction. Okay, thank you so much. Be curious. For any questions, you can always write a mail to arya.priyanka18@gmail.com. Like, subscribe, and follow my YouTube channel, Toxicology on Board. All the best for your exam.